next speaker is Dr. Jourdan Le Saux from San Antonio, who's going to discuss a telomerase activator and its effect on a murine model of pulmonary fibrosis.
Thank you. So it's my pleasure to be here to present our data on this new molecule. I guess that's how it goes. Right. So just a brief overview about telomere and telomerase. So the telomere are these T-loop structure repeat at the end of the chromosome that cap the end of the chromosome. And we know that with age, there is titration of it, which means that when you reach the telomere length, reach a critical length, then it's going to be, the cells is going to go under cellular senescence. So what are we interested in telomere and telomerase activity in lung fibrosis is because there is a bunch of literature that are pointing out that telomere may have a role in IPF, and we saw the talk, we hear the talk on Sunday about it that confirmed that too. So telomere length predict the disease onset. We know there is familial form. There is a connection between telomere length and lung alveolar integrity, and there is the loss of telomerase activity that is associated with myofibroblast differentiation. The telomerase complex is composed of two main compounds. The first one is the catalytic protein, the TERT, and the second one is the RNA with the TR. For the purpose of this work, what we used was the heterozygote mice for TERT. So we have reduced telomerase activity in the mouse. However, those mice don't have the usual phenotype of the TERT homozygote mice. So the Jourdan 510 compound was developed by Jourdan, and it's a novel small molecule activator of telomerase. And they have proven that in vivo and in vitro that this small compound can promote telomerase activation in multiple cell and tissue types. It's also bioavailable when administered by parental route. So we designed this experimental design to try to assess if this little compound can have any effect on pulmonary fibrosis. So we treat the mice 2 days before giving intratracheal injection of bleomycin, and we let them go for 21 days. We also have a subset of mice that we treated with the GRN 163L, and I'm sorry for all this acronym, but and this little molecule is actually a telomerase inhibitor. And the reason why we did that is that we wanted to make sure that the effect we had observed was linked to the telomerase activity and not to any additional function that telomerase can have, especially in relation with the Wnt pathway. The outcome measurement are the classical one. We'll use lung physiology to measure compliance, elastin airway resistance. Of course, we measure telomerase activation. Inflammation was part of the study. And because of telomerase and telomere, we look at cellular senescence. And since we're working with fibrosis, we look at collagen deposition. The first clue that we had that we have something happening in those mice is that we have a better survival of the mice when they were treated with the GRN 510, and it was significant. So we had an idea that maybe something was happening in those lungs. But before analyzing the lung, we look for the telomerase activation in the lung. So it was a whole homogenate of the lung. And by trap assay, we were able to show that on the top panel, that we have activation of the telomerase that was significant. And it was after, even in the 1 week after treatment, and it was sustained throughout the experiment. When we treat the mice also with the telomerase inhibitor, we were able to suppress that activation. And with the southern blot analysis for telomere length, we have a significant, even though very short, increase in telomere length in the mice. So those untreated one means that they are treated with the vehicle, they are bleomycin and treated with vehicle. But then when we look at the lung of those mice treated with the GRN 510 over time, so both group of animals have a decrease in compliance compared to the PBS mice. But the decline in the one treated with the GRN 510 was reduced. And that was correlate with, as you can observe here, with the H&E staining with the less injury in the lung. And then when we did the quantification of the collagen with Pachycereus red, there is a significant reduction of collagen both at 2 weeks and 3 weeks. Now, when we treat with the telomerase inhibitor as well, which is the one in the middle here, I won't say we restored the phenotype, but we didn't, the GRN 510 was not able to protect against the bleomycin injury as well as when it's by itself. And when we look at the collagen 1 expression, we also were able to show that the gene expression was changed. So the change that we observed were at the transcriptional level. We look at the senescence. So the middle one, the panel here is the one when we treat the mice with bleomycin and the telomerase activator, and we did observe less beta-gal staining positive cells compared to the one with bleomycin. And also when we did the staining with MH2A, which is a chromatin marker for senescence, we did also notice a reduction in the staining. That was compensated when we treat with GRN 163L. So when we block the activity of the telomerase, we have a phenotype that look more than the plain bleomycin. When we look at different markers for senescence, also P21, which is the one that is leading with the DNA damage, was the one that's the most significantly reduced, and that makes sense because that's what bleomycin is doing in those mice. So we were protecting that pathway. We also have a dose response. So most of the mice that we were, the mice that we did was at 10 milligram, and then we did one set of experiment with 20 milligram, and we were able to have even a better protection. So we do have a dose effect of that compound. Now the question is, what is the cell type in the lung that may be the target of that drug? Because we observe cell senescence, protection against cell senescence, we have a sense that it might be more the epithelial cells. And then what we did some in vitro study with the small airway epithelial cells versus fibroblasts, treating them with the GRN 510, we were able to show that actually the epithelial cells seems to be the cells that respond to the drug. And what we present here is the telomere length detected by southern blot. And when it's also a dose response, that when we increase the dose of the GRN 510, we have a better protection from, we have increased telomere length. So we are thinking that the telomere activator is actually blocking the cellular senescence, and that way protect from all the downstream effect that we see with fibrosis. So the target seems to be that cells. We need to identify more clearly the mechanism, of course, but we are trying to do that. This has been an effort of many people. I initiated that work when I was still in Hawaii with Dr. Rich Alsop and in collaboration with Jourdan. And we have been continuing this work since then. And I'd be happy to answer any question.
Okay, Claude, very interesting. I have a couple of questions. I'm not sure if I got your data correctly. Did you show that the lower dosing groups had a better survival as compared to the higher dose? And is it true that in the lower dose group, the telomere length was not significantly expanded? That's one question.
Okay, the first question. The significance between the lower dose and the higher dose was not significant because I shrink the scale here. So we are talking about difference in one animal. So the dose had no effect on the survival. The second one was...
length increase, was that significant in the lower dose?
Yes.
It did appear that there was only a...
No, so even at the lower dose, the 10 milligram, we did have a significant increase in telomere length, telomerase activity, and reduction in collagen deposition.
And is it correct that you used heterozygous telomere knockout mice?
Yes.
What is this? I mean, what is the telomere length? Is this the first generation? I mean, I know that if you have a complete
Yes.
knockout, you need to wait for 4 to 5 generations until you get to the stage of
Yes.
cellular senescence in mice because they have so awfully long telomeres. So,
Yes, so that's one of the reasons we use the heterozygote mice, because we wanted, when we started the project, we wanted to show we were able to identify increased activity of telomerase in the wild type. We have reproduced all this data in the wild type, and we showed the same thing. The heterozygote mice that we use were always in the same group of generation to avoid the difference in the telomere length that could have led to difference in the project here.
And in absence of treatment, what was the telomere length in these mice as compared to wild-type mice of the same generation and knockout mice that have a homozygous knockout mice?
So we didn't look at homozygote mice. We didn't do that. We didn't have enough drugs to perform the same assay with the homozygote mice. And we haven't done, we haven't run in the same treatment group, the wild type and the heterozygote, but when we measured the telomere length, they were within the range of wild type.
Okay.
That answer your question?
Yeah, I mean, the question is then, would they already been prone to
No, they were not.
senescence because of the telomere length? And
No.
how does your drug work? I'm sorry.
So one of the things that we did also is that we look at cellular senescence in the PBS mice after 3 weeks, and the level of senescence in wild type and heterozygote mice were the same. So we didn't detect any changes. So the het mice were comparable in the wild type as far as we could test.
Other questions?
Yeah, yeah, I think so. I forgot my question.
I meant, does anyone else want to ask a question?
Could you explain again how you determined that the drug effect was in the,
Epithelial cells?
but you said epithelial cells, but then you said small airway epithelial cells. Is that opposed to alveolar epithelial cells? I'm unclear how you made that determination.
Okay, so when we observe the cellular senescence in the pathology, when we look at the cells, there seems to be more in the alveolar epithelial cells. We haven't.
Which ones?
The alveolar epithelial cells.
Alveolar.
So we haven't followed up on that yet in defining exactly what is the cell type in vivo that respond to the drug. What we did is at the time when we initiated that study, I didn't know how to isolate alveolar type cells. So we used a cell line, and it was a small airway epithelial cells for the in vitro study.
I see.
It was a convenient cell line to use.
Okay.
Okay, sorry, it wasn't clear.
So have you looked at off-target effects of this of this activator?
Yeah, so such as cancer. So there have been a study going where the mice have been treated with the telomerase activator for over a year, and but hasn't been conducted by us. It has been conducted by Jourdan when they were testing that molecule. And they used different mouse models, and I think there is a publication that came out like 6 months ago about that, and those mice didn't develop any side effect, which is sort of surprising, I know.
I'm sorry to come again with a question, but I think this topic is quite interesting.
We can talk after if you want.
If you do an immunostain for telomerase in mice in murine lungs, where would you find it?
If I do what?
An immunostain.
Oh, like the fish one?
Well, whatever. Immunohistochemistry for telomerase in murine lungs. What is the cell type that shows the expression? The telomerase.
Yes, I understand telomerase.
In which cell would you find the telomerase?
Well, that's the problem. That's one of the questions that's been raised over and over when I try to get that funded, is that the mouse level of telomerase activity is high in every cell type. In every cell type.
Okay.
There is no, in human, you don't have so much telomerase activity, but in the mouse you do. So that's one of the reasons we use the heterozygote mice.
Okay.
Moises?
One brief question. If I understood well, following the question of Marvin, you have your in vivo result, and then you take a cell line of bronchial epithelial cells and an embryological fibroblast.
Yes. No, the EMR 90 also a cell line.
It's not, yes, it's a cell line, but for fetal lung, I think. It's
Yep.
not from adult. So you demonstrate that in a fetal fibroblast, your drug have no effect on telomerase, but on the cell line of this bronchial epithelial cell, you have the effect. That is, well.
So this piece of information, we did that to try to target what are the cells that might be responsive to the drug. Because that's what we observe with the data with the cellular senescence. We only see the cellular senescence at the epithelial cells level. So we wanted to find out if it was just something with the mouse model or if we had something the same in human cell line.
It is difficult to understand for me, but I am a real ignorant in this, is how, if you put a parenteral activator of telomerase, you have an effect of an epithelial cells and not on fibroblasts or inflammatory cells. I am
Okay,
not
for the
sure
inflammation, this is a good question for inflammation because I think one of the problem we have in the mouse model is that when we test some new compound, they may have an effect on inflammation.
Yes.
So what we have done also in a very limited number of mice, so these data are very preliminary, is that we treat the mice 7 days after bleomycin and not before. And we did observe the reduction in collagen deposition. It wasn't as striking as when we treat in the current protocol I'm presenting, but we did have reduction of collagen deposition, even if we don't touch the inflammatory response to bleomycin. So that's what encouraging too. And we observed that in the heterozygote and the wild type.
The last question, brief. How do you link the increase of the telomerase activity in epithelial cells in vivo and the decrease of collagen few days after?
Well, that's the grant I'm going to submit. Because we have observed also with another model, the caveolin-1 deficient mice, that if we were able to block cellular senescence in the epithelial cells, we were not seeing the collagen deposition downstream. So we are thinking that the cellular senescence in the epithelial cells might be one of the triggers of the cascade that is going to lead to collagen deposition. Remember that the cellular senescence are releasing in their microenvironment a lot of cytokine and growth factor. Among those cytokines, there is interleukin-1 beta that has been shown to activate the fibroblast. And there is a recent publication, I think a couple of months ago, showing that if you treat actually human bronchial epithelial cells with TGF-beta, with 2 nanograms per ml for 48 hours, you induce cellular senescence on those cells. And when you treat fibroblasts with the supernatant from those cells, those fibroblasts become myofibroblasts and produce collagen.
Thank you very much.
Thank you.